I hate that word. I don't. That's an annoying.
Because most of the thing is water. What's that? Oh, no.
Yeah, it's having trouble going live to YouTube. Yeah, like it loaded the bar and now it's stuck on a white screen. Ugh.
She doesn't see this.
Yeah, sorry. YouTube is just taking a minute. Okay. Yeah. Hopefully it should load soon.
Oh, it's
okay.
working. It's working.
Kind of. Hang on. There's more progress. Okay.
Progress. Oh, it's muted.
Okay.
All right, nice.
All right. So we're all good on YouTube. So Justin will go ahead and introduce you now.
All right. So good morning, everybody. Welcome to our final day of NVTI presentations. Here to start us off today, we have Patricia Miloslavich, a retired professor and current executive at the Scientific Committee on Oceanic Research. Patricia, if you're all good to go, you can take it away.
Hello, good morning, Debbie. Good morning to you. It's midnight for me. I am now in Australia. And thank you very much for the invitation. So I'm going to be talking to you and I'm going to start sharing my screen about the lessons learned after at least 40 years of practicing marine science throughout my career. So I'm going to be sharing my screen and you let me know if you can see the presentation.
Yep, your presentation's all good.
Oh, Tony.
Perfect. So I've entitled this talk A Journey Through Marine Science, Lessons Learned from 40 Years of Practice, and I hope you enjoy it. So where to begin? We begin in the 60s. That's when I was born. And as you can see in that picture, I loved the ocean from a very early age, as I would like very much to go to the beach and take samples of water and sediment of my bucket. So later on in the 70s, I was quite inspired as a teenager by, as pretty much anyone of my age, by Jacques Cousteau, the French explorer and technology developer, who had this ship, the Calypso, and he traveled around the world making lots of discoveries and then doing a lot of communication, writing books and making videos and movies and so forth. And he came actually to the Venezuelan coast, which is where I lived. I am a Venezuelan and was living there. He came in 1979. I was still in high school, so I did not participate in this expeditions, but I heard about it and it was really, really interesting to learn that he, my hero, was coming to Venezuela. So that was in the 70s. what happened in the 80s? In the 80s, I finished high school in 1980. And what do I study at the university? I always wanted to study marine biology. So I went for marine biology, and I just registered at the university, Simon Bolivar University, from which I became later on, after many years of working there, a retired professor. And my first contact with marine biology at the university was through a professor here, a very old picture of him, Paulo Penchasade, who had left Argentina because of a military regime and dictatorship going on in Argentina in the mid 70s. He came to Venezuela and started teaching marine biology at the Simon Bolivar University. And he was offering some kind of research project in sea cucumbers. And I said, well, I don't know anything about sea cucumbers, but they're marine, so let's go for it. So we started working in marine biology. That was my start. And from there on, I pretty much moved to the study of gastropods, marine snails. So in the 80s, still when I was finishing my career, we had to do a research project of a 1 year duration that was a thesis. And I worked in this family of snails called vermitids because they look like worms. And they're very interesting because they attach themselves to the substrate and never move ever again. So wherever they fix, that's where they're going to live for life. And these snails were very interesting because they have separate sexes. So you have a male and a female, and still fertilization was internal because male produced all the sperm, put it in an egg capsule, and threw it to the water where it was collected by the mucus thread that the female used for feeding. So the two species lived in very different places. One lived in the channel of a power plant. That's one on the left. And the one on the right lived in a beautiful location at the national park. They had very similar development. It was very interesting because we discovered that they brood the egg capsules. They put eggs inside egg capsules, and the egg capsules are brooded in their mantle cavity until they hatch as little snails, which then attach to the substrate and then start making their shell inside that substrate. So when I finished my career, started working, and then I started a PhD. And for a PhD, I moved to Canada. I got a scholarship at the University of Quebec at Rimouski. And I started working at this university with Louise Dufresne. She was my advisor. And we started working on the reproduction of a snail, Buccinum cyanum, that came from the Saguenay Fjord, which is a tributary of the large St. Lawrence estuary. So this snail was very interesting. It was found on relatively deep waters, well, beyond 50 meters depth, 50, 100 meters depth. And the question here, the scientific question that I wanted to test was because the size of reproduction was quite variable. There were little snails, large snails, but they still all reproduce. So the question was if the larger snails would have a higher fecundity, because this is a fishing resource. And if people fish them and they fish only the big ones, so what would be the consequences for the population? And we found out that larger females did indeed produce larger egg capsules, more eggs per capsule, and more juveniles per capsules. But the juveniles at hatching were no different, either in size or in terms of their biochemical quality, as those that came from smaller females. So this was quite interesting because even if the production was higher, still the quality was pretty much the same. So the 90s, I continued to work after my PhD. I came back to Venezuela and started working at Simon Bolivar University as a full professor. And for many years, for the remaining years of the 90s, I continued to work on reproduction of marine gastropods, studying how they reproduce and the different strategies they have to provide their young with extra embryonic food sources. And there were many things that the females did. Some of them put extra protein in the egg capsules. Some of them put extra eggs in the egg capsules that were used as nurse eggs by the ones that did develop. Some of them actually all started develop, but then little snails started eating to each other in a way of cannibalism. And so it was quite interesting. And the question then was, what was the reason for which some of these eggs did not develop and became nurse eggs? And then I studied the biochemical content of these nurse eggs and found that actually they had lower content in protein, sugars, and lipids than normal eggs that would actually develop. So they started disintegrated at a very early stage and then were consumed by the ones that actually did develop. In the 90s, I did some reproduction of my own, and I'm very proud to say that, well, in 1991, when I was just starting my PhD, I had my first son. Then in my last year of the PhD, I have my first daughter, and then 4 years later, I had my second daughter. So 3 kids, all of them very fond of water activities, both beach and freshwater. So while I started raising kids, I continued with my professional career. And in the 2000s, as an established professor at the university, I had several students and colleagues that worked with me at the lab that I ran, the marine biology lab at Simon Bolivar University. One of them was my good friend, Elizabeth Hook, who worked with the gastropods or snails associated to seagrasses, the Thalassia testurinum, which is the turtle seagrass bed. And she found in an area, the Moroccoin National Park, around 26 different species of micro gastropods. And the number of individuals per square meters, which was usually around 1,000 and 5,000, which is quite a lot, but that were the normal range. But this number reached almost 45,000 after a flood event that all the seagrass was washed with fresh water due to heavy rains. And it took several months to recover. But when they recovered, one species in particular had an amazing demographic explosion and produced had densities of 45,000 per square meter. I also work with Anna Carina Carbonini, and she worked on the reproduction of a family called the calyptrades, which attach to other shells and brood their egg capsule as well. And she also worked on the regeneration of the siphons of some bivalves after they were nipped by fish. In the 2000, I also made a postdoc at the University of South Florida, and here I worked with John Lawrence on studying the protein and carbohydrates content of larvae of one sea urchin, the one you see on your left. And we submitted the larvae to 2 diets, a low, very low content diet in terms of microalgal cells that you feed them, because you feed this echinoderm algae larvae with algae. And so in one, what I call the low diet, I really starved them with a very small quantity per liter, and in the high diet with a very high quantities. And we found that the low diet, the larvae had a very low content of protein and carbohydrate per larvae, and also did not complete their development beyond the 4-arm stage. So that was my postdoc at the University of Florida for about a year, and between 2004 and 2005. I also had a student in those years that she came back to that power plant channel where I had done my undergraduate thesis, because in the 80s we had observed that there was another vermetus species, but in very low density. So we came back, this was 20 years later, to see what was going on there. And we saw that massive formations of that snail, and we were impressed with what had happened there. And she looked at the genetics of that one, and she saw that there were 2 different colors. There were the black ones and the oranges one. And it resulted that the black one, which is the one that I had seen 20 years before, was a Caribbean endemic species. So it was, it belonged there, but it had reproduced in a very high quantity. But we had received in the last 20 years an invasive species that came from the Pacific Ocean. So it crossed through the Panama Canal probably attached to some ship, and then, or the larvae in some ballast water came to the Caribbean, established, and then fixed itself on this channel. Also in 2000s, because I was doing so much biochemistry on the protein content and of the embryos, I had the question of what determined the hatching size in gastropods when they have these extra embryonic food sources I mentioned at the beginning. And I studied several species around different 20 species. And we found that regardless of the size of the species, so very tiny, tiny species or very, very large species, they would, what determined the the hatching size was basically the amount of protein that was in that either the nurse eggs or the intracapsular fluid or through cannibalism that the embryos had at their disposal while developing. In the 2000s also, I was involved in this beautiful project, which was an initiative of the British Council Darwin Now Network. They called it Darwin and the Adventure, and they wanted to recreate the travel of the beagle, but now, well, 200 years later, with still on a sailboat or a tall ship, but using modern technology. So we studied the feasibility of using a tall ship to take samples of plankton, or how could we do and recreate, at least from the marine perspective, the same that Darwin did many years ago. So this was just a test, and my understanding was that a ship was going to be built, but at least it did not come to South America as we have planned. Then when the 2000s started, I said this was a time of many projects, because at the same time, in 2000, the Census of Marine Life started. And the Census of Marine Life was a project to study the diversity, distribution, and abundance of marine life at the past, in the present, and in the future, from the coast to the open ocean to the deep sea, for all taxa, from microbes to whales. And I must mention here the special role that these gentlemen here on the left had, because Fred Grassley, who was a professor at Rutgers University, he was a visionary, and he was the one who envisioned this program. And he talked to Jesse Oswald from Rockefeller University and the Alfred Sloan Foundation to try to get the funding to get this started. And I was engaged by Juan Manuel Diaz, a colleague from Colombia who worked at the diversity office of the INVEMAR. And he pointed out that I could help put together information from Venezuela. And then I ended up working for 10 years, putting together the information first for Venezuela, then for South America, for the Caribbean, and then globally. So the census was about putting together a program that had 17 projects. More than 2,700 researchers were involved from 670 institutions, more than 80 nations, 10 years, and 13 international and national or regional committees. So the projects were coastal. They went for the coastal ocean, the polar oceans, the pelagic regions, the deep sea, and all this information was compiled in the Ocean Biogeographic Information System, which is now under the umbrella of the Intergovernmental Oceanographic Commission. So what did the Census of Marine Life discover? That the marine life is more diverse, more connected, and more altered than we previously thought. And these maps here show the biodiversity hotspots, meaning the places that have the more diversity, marine biodiversity in the world. And you can see it's around the waters around Australia and Japan, all what it's the Indo-Pacific Ocean, and also quite a hotspot in the Caribbean, and then in the west coast of Africa, in Southern Africa. And then on the right, you have the graph that show all the lines and that show how the oceans are connected through the animals. The animals, more than 43 species of animals were tagged, around 20,000 animals. And the census of marine life followed them throughout the oceans and saw how they migrated from coast to coast, crossing all the Pacific Ocean, surrounding the Antarctica, going from the Antarctic to South America, from the Antarctic to South Africa, from the Antarctic to Australia and New Zealand. So it was quite impressive to see all this migration happening. The census of marine life was also through the census, also the first time that we were able to do an inventory of what is out there. And the known diversity showed us that 50% of all diversity is pretty much composed, at least in terms of big animals, invertebrates and vertebrates, by crustaceans, mollusks, and fish. The rest, the other 50%, is pretty much all the rest. And this does not include the microbial diversity, which we know today is very, very high. But still, our knowledge was quite superficial because, as you can see from the graph on the right, the red represents the areas in which we had data. And data was pretty much restricted to the very shallow zones up to 1,000 meters depth. So very poor information from the deep sea. And also, we found that endemicity, which means species that are found nowhere else, was very high in the south. The census overall described more than 1,100 species and had more than 5,000 to be described at the time it finished in 2020, in 2010. The census also provided the first marine biodiversity distribution maps in the Caribbean and South America, showing, again, where the hotspots of where the diversity is more concentrated, which is what you can see in these maps. The red parts are the ones that have more reported diversity. And you can see here in the Caribbean, the fish records, there are fish records everywhere, and these are pretty much at the surface level. But if you go to the graph below, which are the deep sea records, they're very restricted in area. After that, when the census finished in 2010, I started to, from there to build. Once we have the foundation of what is out there from the census of marine life, let's now look with more detail to the different communities. So we started a project called Marine Biodiversity and Biomass Assessments on Coastal Ecosystems in South America, which was monitoring rocky shores at the regional scale for several years at 150 sites using standard protocols. All this monitoring was done for science and education and to help inform policy to establish management practices. All this data was contributed to the Global Biodiversity Information Facility. The protocols were published in the Ocean Best Practices, and the papers showing the results, how all these groups are distributed in coastal rocky shores were also published, and these are some of the maps. 2010, then I also started move from the rocky shores to go deeper because I had the opportunity to go as a scientist on board the Nautilus, the ship from the Ocean Exploration Trust, an exploration vessel, to the 2013 expedition to the Kikamjeni underwater volcano in the Caribbean. And it was really a life-changing experience because I would be, I had my shift and had to broadcast what we were looking. And one of the nights I was on shift, I think it was probably 4 o'clock in the morning, we discovered through one of our ROVs, remote operated vehicle, deep community of mussels in a seep. So this was very interesting. We collected some of these mussels, the giant mussels. They measure around 20 centimeters each. And each of these mussels inside had further, as a parasite or a host living there, they had a worm living there. So it was very interesting to live this experience on board the Nautilus. After the Nautilus, I came back home to continue my work at the university after the expedition, and I had another, this was a PhD thesis by Carolina Peralta, in which she worked in a species called Voluta musica, which is a species that was considered as vulnerable, but there was in the red books of conservation. But because there was a lack of information, there was not clear in which status to put it. So we provided information on its fecundity, and we discovered it was threatened by several problems. One was overfishing, not of that particular species, but the species of which this one fed off. So this one lived in a bank of bivalves, some very probably very tasty bivalves. So this species would eat those. And when the trawling fisheries came to trawl on that resource, they took the volatids as well. The second threat was that they suffered from imposex, which is a phenomenon that happens. It's male imposition, male character imposition on a female, causing infertility. And this was caused by a chemical that is released by the paintings used on the fishing boats. And the third problem was that the fecundity of the species was very low. So with all this information, we were able to provide a better category under the Red Book, which was then can be used for better management practices. Also, I had a student working on the effects of imposex, but now on another species, and trying to find out throughout all the Venezuelan coast from west to east, where were the highest concentrations. So he went to the marina to sample, but also to the ports. And he found out very interesting that the content of TBT, which is a component that causes the imposex, and snails suffering from imposex was much worse in private marinas rather than ports. And this was probably to be expected because international laws for ports are more strict. So this TBT is banned by international laws. But if you have just a small vessel and you don't leave like the national waters, you are not going to be as controlled as those that live from ports. I also had other students, Jose Antonio Faria, who worked on the effects of rivers on rocky shore assemblages, Nicida Noriega, who works on the effects of upwelling on reproduction and biochemistry of sea urchins, and Maria Gabriela Neves, also on the impact of the trawling fisheries and minimal catchable sizes of the resources that of the place I mentioned before. Now, as the 2010 was finishing, and well, now that new decade, 2010 to 2020, in the mid 2000, like around 2015, I was hired by the Global Ocean Observing System to go to Australia and study and build up the global networks to have a biological observing system. There was a global ocean observing system. There was a framework for ocean observing that had been published in 2012, but it had been agreed by the community that the ocean observing system needed more than just the measurement of physical and biogeochemical variables. So it was my task to build up the networks and understand what were the biological essential ocean variables that needed to be measured. And for that, we came out with NICBACs and what I just mentioned to study, well, how do we measure how do we determine what is essential to measure in a monitoring program? And to have sustained observations, ocean observations, we said, well, we need to respond to society. What are the societal needs and the international obligations that need to be addressed? So we reviewed international conventions to identify the requirements. We look at what observing programs are already out there to see, the low-hanging fruit. What are they measuring that can address the requirements from society? And then we prioritize those in terms of based on feasibility and on their impact. And then finally try to provide solutions. So we came out with a list of drivers. What do we need to do? And what are the main problems that we need to address? So the main drivers are environmental quality, threat prevention mitigation, food security, biodiversity conservation, sustainable use of biodiversity, capacity building, sustainable economic growth. And then how do we, with these, address the problems which are loss of habitat and resources, climate change, pollution, eutrophication, coastal development, solar waste, and so on. So with these problems and these requirements, and we came with the solutions, and we us address the information that policymakers need at the minimum are information, basic information on functional groups, and also some information on habitat state. Functional groups, what is the diversity of and biomass of microbes, phytoplankton, zooplankton? How does it change through time? How does the distribution and abundance of benthic invertebrates, fish, and turtle birds and mammals change through time? And also, how do the cover and composition of hard corals, macroalgae, seagrass, and mangrove, all of these ecosystems are building ecosystems for a lot of other organisms that live in there. How does this change through time? So while I was working with Goose, we also integrated with the Marine Biodiversity Observation Network, the MBON, and built the pole-to-pole program to develop capacity in data collection, data formats, data publication, and data science tools. And we organized several workshops to actually bring together people from all of the Americas from pole to pole to work together on this. And then in the 2020s, well, as a retired professor now and retired from Goose, I started working for the Scientific Committee on Oceanic Research, SCORE, which is where I work now, which is where from I can now serve the global oceanographic community in a much broader way throughout all the disciplines of ocean science. So SCORE, it has a mission of addressing global and multidisciplinary ocean issues and conducting research through working groups and projects, solving methodological and conceptual problems and building capacity. And I'm gonna show you, because I think this is much better explained in a video that explains what are SCORE activities, because SCORE basically has working groups, research projects, infrastructure projects, capacity development activities throughout all ocean disciplines from biology, physics, biochemistry, chemistry, geology. There's a lot of partners involved for each of these projects and the working groups, and of course the sponsorship of many organizations. So I'm going to show you a video now, for which I need to escape here just a moment. I'll put the video, share the sound. And there you go.
The Scientific Committee on Oceanic Research, SCORE, is an international organization promoting international cooperation in ocean science. The mission of SCORE is to address multidisciplinary ocean issues, plan and conduct oceanographic research, solve methodological and conceptual problems, and develop capacity for ocean science. SCORE promotes equity, diversity, and inclusion, supporting students and early career scientists.
We can't currently see the video.
Since 1957, SCORE has shaped modern oceanography by planning and coordinating several large-scale ocean research projects and establishing 165 working groups involving thousands of scientists from all continents. Currently, SCORE activities contribute to the UN Decade of Ocean Science for Sustainable Development. SCORE's current research projects are focused on marine biogeochemical cycles of trace elements and their isotopes, on ocean sustainability in the context of global change, on the biogeochemical physical interactions between the ocean and atmosphere, on ocean soundscapes and the effects of sound on marine organisms, and on the Indian Ocean processes and how they affect the Earth system. SCORE supports infrastructural projects which tackle methodological, observational, and data and information related issues. The current infrastructural projects focus on ocean carbon, Southern Ocean observations, harmful algal blooms, and the effects of multi-stressors on the marine biota. SCORE currently has 19 active international working groups, which are developing and standardizing novel experimental designs, tools, and databases to understand a variety of physical, chemical, biogeochemical, and biological processes. Related to physical oceanography, SCORE working groups are producing a quality-controlled ocean database of surface temperature profiles, translating optical measurements into particle content, aggregation, and transfer, analyzing and modeling floating litter and its oceanic transport, and analyzing ocean turbulence observations to quantify mixing. Related to chemical oceanography, SCORE working groups are developing a global network of ocean time series of dissolved nitrous oxide and methane, modeling chemical speciation in seawater, and facilitating an intercomparison of iron models. Related to biogeochemical oceanography, SCORE working groups are measuring essential climate variables in sea ice, reconciling ecological, biogeochemical, and model estimates of respiration in the Mesopelagic Ocean, developing and observing air-sea interaction strategy, and coupling the ocean ice-atmosphere biogeochemical processes. Related to biological oceanography, SCORE working groups are integrating plankton observing sensor systems to existing global sampling programs, studying the dynamics and sensitivity to climate change of eastern boundary upwelling systems, standardizing active chlorophyll fluorescence to measure global marine primary productivity, developing an atlas of zooplankton biodiversity based on DNA metabarcoding and sequence databases, coordinating the global research assessment of seagrass systems, developing a roadmap for a global approach to deep sea biology, standardizing coastal and nearshore demersal fish visual census techniques, and developing novel experimental designs and tools for the study of mixotrophy in the oceans. SCORE promotes and supports networking and training activities across all ocean science disciplines. Through its capacity development programs, summer schools, and regional graduate networks of oceanography, SCORE aims to provide the skills and knowledge needed for oceanographic research, along with the opportunity of shipboard experience. From the field to the lab, from data analysis to global synthesis, SCORE develops capacity in ocean science at every step of the way.
Okay, so I'm back. Can you hear me?
So yeah, we can hear you, but we weren't able to see the video. We were able to hear the video just fine, but the visuals weren't there.
Oh, I'm sorry about that. I'll share a link of YouTube. It's sorry, but because we tried it before and it worked.
Yeah, it did. And honestly, we don't really know why it's not working now, considering we did try it before, but I'm very sorry about that.
It's okay. At least you heard what it's about. And I can share the link of the video later. Okay. So, well, my two slides to finish. Well, what are the lessons learned? And the first one I want to show with this graph here is the power of collaboration in scientific production. In the graph of the left, you have, and I used my own experience in this. In my first years, when I started, publishing in the late 80s and then the 90s and then 2000, it was just me and one or two more authors or co-authors. So we would have like a publication a year, maybe two publications a year if we were really lucky. But when you start the collaboration and you have, you involve 10 people or more in an article, and you start, really collaborating in science, You can see how that is really a powerful way to actually start publishing more. So on the right, then you see the average number of co-authors on the x-axis, and then on the y-axis, the number of published papers. And the more co-authors I had, it was more papers I produced. So this is the first lesson I wanted to show. But then I have like other words of advice, especially as you are at the high school level finishing. There's an academic career to be successful requires a lot of work. Of course, you have to be smart and study a lot. So that's not the problem. You have to have interest in what you do. If you are interested in what you do, you will do it with pleasure. And it makes a whole world of a difference to do things when you're interested in them or you're just doing them because you have to. So interest, find something that is interesting for you and go for it. The second one is perseverance. Even if you're interested, you will find many times rocks in your way, and it will be hard, and it will be time consuming, and sometimes it will be very frustrating. So you have to persevere because you will be able to succeed at the end. Other words of advice, well, be patient because growth will come, knowledge will come, experience will come. The more you practice, the more you do things, the better you will become. But all of that requires that you are patient. You were not born having learned things. You have to learn throughout the way. The second one is to develop skills, whatever they are, other languages, programming, computer coding, drawing, software, anything. Whatever you learn, any skill that you learn will be a benefit later on in your career. Also, to be aware of opportunities. The Census of Marine Life came, and it was because I was there ready, and I said, yes, I'll do it. So when you get invitations to collaborate, they say, yeah, you have to have an open mind and say, yeah, I'll do this, because those are opportunities that are life-changing. Also, to be part of teams, because your voice will have influence, and by being part of teams, you will also be able to connect more with other people, which takes me to the next point, which is networking. This will allow you to connect, to collaborate, and to share with others. Science is much fun when you do it with other people, and it's also not just much fun. You do it much better because you have this intellectual exchange that is very enriching for science. Then also to communicate, try to reach out. Whatever you learn, try to reach out. Of course, you have to publish because that's our obligation as academics, but go beyond that and communicate your findings to the general public. Keep improving, continue to update. An academic career never finishes. You are studying all the time. Keep learning skills, you keep improving, you keep learning, you keep updating. Then you have to work hard, of course, but you have to enjoy as well. You only live once, and you're only young for some years. And so just enjoy, do sport, do outdoors, have a hobby, do things that enrich also your mind and your soul and your heart. And then rely on and be supportive of your family, your friends, and your colleagues. They are the ones that are going to be there with you. They are the ones walking this path with you, and they will be there when you need them. So I just wanted to thank, of course, you for the invitation. I want to thank all the institutions that have made possible for me to have this very enriching, fun, and interesting career. All the programs I work for, the funders, the cohorts and cohorts of students, more than 25 years of teaching that I had at the university, and also in my international courses, my colleagues, my collaborators, all those people who inspired me, all those who helped, and all those that brought me here. So thank you very much, and I look forward to your questions.
Hi, so because we got kicked so many times, we don't have access to all of the questions anymore. So if it's all right with you, it looks like you're going to have to read your own questions and we won't be able to read them out to you just because we can't see them.
Okay, I have 7 questions here. One of them is, while you're doing experiments, do weather events change how you collect and analyze the data? Does water quality change significantly during those events? Well, yeah. While you're doing field work, well, first we try to plan field work to get ideal weather conditions, especially if you're going to look for live animals. But if you're on a monitoring program, you have to monitor at any condition. You just do it. And then, of course, water quality will change during those events. And you will see the consequences on the marine life, either immediately for those that respond very quickly, or in the long term if there was some kind of disaster. Is there ongoing data collection to compare to the census data to see if there's change? Well, yeah, for some groups, like zooplankton, monitoring has gone for more than 80 years, and all the data is still being uploaded. There's still rocky shore monitoring, there's fish monitoring. So basically, the monitoring programs today pretty much measure all of the variables that I mentioned before. The issue is that we are not monitoring everywhere, and that's the gap that we need to fill. We need to take monitoring everywhere and do it globally and in a way that is comparable. So if you can compare results, then you can do global analysis. The data, at least for marine biodiversity, is being uploaded in the Ocean Biodiversity Information System, OBIS, which I think I mentioned is under the umbrella of the Intergovernmental Oceanographic Commission. It's based in Belgium. In Belgium, in Ostend. Is there a place where people can access the data that was accumulated by the census? So yes, so just the biodiversity data, you can find it in OBIS, and you can also find it in many of the places. So you can look at the papers that were there. There's an impressive collection in the PLOS One journal, which puts together the global synthesis from literature sources, not from actual field collection. And you can look into that, and I can send you the link to the collection later, along with the link to the video so you can watch it. We can see the video. Okay, sorry about that. These partnerships are awesome. Yeah, without partners are essential for science to move forward. For many reasons. First, collaboration, as I mentioned before, is enriching from the academic point of view, intellectual point of view, but also we avoid doing the same thing twice. When you collaborate, you put together several brains to work and you do something well done from the very beginning. People can have the same idea in different places, and this happens, and you can have parallel research going on in different parts of the world. So that was very common in the past, but today, with all these communication tools, it's less likely, and it can be avoided because people can communicate much easily. So take advantage of that. How does climate change affect these studies? Well, climate change affects everything. Not just the studies, because, well, you have a monitoring program, you have instruments in the field. Climate change may not just, the instrument may not just read the changes, but they may be affected by the changes if there's a severe storm or a big upwelling or a natural disaster, hurricanes, that you may lose actually the equipment. And well, of course, if you're going to the field and the weather conditions due to one of these extreme events due to climate change is going on, but you can't go to the field and then you will miss the data. So those are the questions that I have. Oh no, there's another more here. Do politics impact these partnerships? In occasions they may, yes, in occasions they may. Scientists try to be, let's say, more immune to politics in the sense that we collaborate with each other regardless if our governments don't like each other. But of course, I think no one in this world can escape at the end politics. I put as an example the Intergovernmental Oceanographic Commission, which is a governmental organization, and signatories are more than 100 countries that have access to the ocean. And of course, there's a lot of politics there, but there's of course a lot of diplomacy. And GOOS, the Global Ocean Observing System, was a program under the IOC, and still it was a program that was approved by all member states, which meant that even if they have their differences, they approve that this program goes forward because it's for the interest of all countries. Politics also decide where funding goes, and this is where society can make a big difference. Because if society makes pressure on their governments to address needs like climate change, like the need for funding for science, like the need for more schools, like the need for more teachers, more research, more funding to research, more discoveries, because there's a need. When there's a need and the need is put forward with enough pressure to politicians, then there will be the political will and the funding for this to happen. But society needs to play a big role in this. And that's why it's so important that us scientists communicate well our findings to society to convey the message of how important it is to understand these natural processes. How many scientists and students were involved in taking the census of marine life? So it was 2,700 people involved in the census of marine life, quite globally, so quite extensive. Did COVID impact any of the research? That's a very good question. COVID did impact research in many, many ways. It impacted some people more than others. Who did it impact more? Those that went to the field and had to collect samples because ships were not going out. You had to stay at home. Those that were not as affected were those that work in their computers doing models. Those that work in the lab were also quite affected because universities were closed, labs were closed. If you had experiments running and you did not have access to your labs, you lost the experiment and very precious time. Also, people with young children, early careers, mid careers, who had young children that were not going to school and people had to work from home. So imagine 2 kids doing homeschooling, each one in a computer, both parents in a computer all the time during working hours. So young scientists, your early career especially, felt that they were not balancing enough their professional life with their personal life, and it was very stressful. Equipment that was at the field and could not be maintained was either lost or the data from them was lost. So COVID did impact a lot. It helped. It was not all bad because we can say that connection advanced and made a quantum leap with COVID because people were connected remotely. So they could connect, talk to each other, solve issues that normally they would have waited to get together in a meeting or in a conference to talk to, but they just resolved things more quickly. SCORE, for example, our working groups are supposed to meet once a year. What they did was they So we have 19 working groups. So instead of 19 meetings, we had more than 100 meetings because they met all the time. Of course, not for the same duration, but they met more often and could solve problems in a more, let's say, periodic way. What can we do to improve the conditions of our oceans? We can do many things. One of the things we can do, the first one is to make pressure to our policymakers, to our politicians, that we want a clean ocean, we want a transparent ocean, we want a sustainable ocean. And all these changes need to come not just from the top down, that is laws by the politician that permeate down, they need to come from the bottom up. So we also need to make different choices. We need to choose what do we eat, how much is our footprint, our carbon footprint on this planet. So try to improve and be ourselves more green, more conscious, recycle more, use that mantra of reduce, reuse, recycle as much as we can. And we will eventually be have a cleaner ocean. How is sea ice measured? What do marine biologists look for in sea ice? Well, in sea ice, you can look for many things. First, how do you measure it? You have instruments and you make cores. So you put yourself and your instruments on top of the ice shelf and then start making a core down, like a cylinder that goes down, opening a hole. And then you extract all that cylinder of ice. You keep it frozen, of course, otherwise it will melt and then you will lose information. And look at different elements that are included in your sea ice that will indicate different conditions in the past and how long ago this happened. In the ice, you can also find microorganisms that are frozen, and this will also indicate what was the biodiversity at each of these different levels within the sea ice. So it's very important to understand past conditions in the planet. Did any of your children follow you into the field of science? Well, my children have grown up. My son, well, they were born in the 90s, so they have between 22 and 30. My older son is a mechanical engineer, and he did a master on renewable energy technologies, and he's now working with drones and how drones can map different surfaces. So it's kind of scientific in a way through engineering. Then my middle daughter is an artist. She is a graphic designer, and the young one is going to go into science as well. They came with us to field trips. My husband is also a marine biologist, so that makes two of us. So they they must have it in their genes. How did you manage to find funding for snails? They are not very glamorous. We are curious. Well, they are very glamorous, and actually within marine organisms, marine snails or gastropods or mollusks in general is the second most biodiverse group in the planet after the insects. That makes them a very interesting group because they're interesting not just because, well, they're very diverse, because they are everywhere and they have so many roles in the ecosystem. You can find them in all ecosystems, different roles, from herbivores, carnivores, detritivores, eating dead things, and contributing very much to the balance of the ecosystem. And in addition, because they're so diverse and snails are can have beautiful shells, there's a whole community of seashell collectors, which, well, that's not very ecological thing to do, but they produced an amazing amount of work on biogeography by collecting snails and describing the locality. So a lot of information was provided from many years ago, more than 100 years ago, by shell collectors. And I think that was the last question. And if I look into the chat, there's also a few questions. One, well, the one about inspiring my kids, that's done. Do you prefer teaching or research? Would you want to go back to teaching? Well, I never stopped teaching in a way. Here I am. I retired from teaching, but I still continue to do some talks eventually, like this one. And until last year, well, not within the pandemic, but just before the pandemic, I was already retired from the university, and I did give a course in Argentina and in another in Colombia. So I enjoy very much teaching. I also enjoy very much research, and now I am enjoying very much helping others do their research. So I think you go by phases in your life. Has the global pandemic hindered data collection on any of the projects? Well, for the score particularly, yes. As I said, ships were not going out. Lab work stopped. So people pretty much in these 2 years work in the data they had and produce many, many papers. So in the last 2 years, the SCORE community produced more than 160 scientific papers, which is comparable to levels of publications before the pandemic. Is snail shell color determined by genetics or by what they consume? Let's say that's an interesting question. It's by a mixture. So snail, it's genetically determined. There are varieties, but for some species, not for the shell itself, but for the soft body, what they eat can come into the tissues, especially if they eat, for example, some eat phytoplankton or macroalgae. The chlorophyll containing the chloroplast will be absorbed by the tissue, will actually not be absorbed, will go to the tissue. And some snails then will have the capacity to do a little bit of photosynthesis in their tissue. So that it's quite an interesting phenomenon that we see in other also groups in nature. And I think I've done it with the questions, and both from the chat and from the Q&A.
All right. Yeah. Thank you so much. Again, we are so sorry for all the tech issues we were having and all the internet troubles, but you did great. Thank you so much for taking your time with it and just helping us through it all. We appreciate that so much. Your presentation was great. We loved it so much. And we really do hope that you come back next year and think about it, because this is definitely a presentation that is very beneficial.
Well, thank you for the invitation, and well, I'm sorry you didn't see the video, but if you, I can send you the link, and if you are, I mean, you can share it with the people who were connected. That would be great.
Yes, we will definitely do that.
Yeah, and I can also share, if somebody was interested, I can share the link to the Ocean Biographic Information System, and also that collection of papers in +1. If they want to read more about the census of marine life, that would be.
Okay, yeah. We will definitely make sure those are connected to the YouTube video that is going to be posted soon after this for anyone who is interested in those. Yeah, thank you so much.
Thank you. Bye-bye.